Welcome to the Endpoints podcast, presented by the ALS Therapy Development Institute. I'm Jonathan Gang. We need more, likely many more, effective treatments to end ALS. Progress in ALS research has been too slow for too long, but in 2024, there are signs of hope. The past few years have seen more treatments for ALS in clinical trials than ever before. While most drugs that reach trial fail in any disease, the more experimental treatments that are in trials with good preclinical evidence, the more likely there are to be successes. Recently, we have even seen approvals for new ALS treatments, such as tofersen, a genetic treatment for SOD1-related ALS sold under the brand name Qalsody. Dr. Fradette was one of the people behind this milestone. As the VP head of Neuromuscular development at Biogen, she oversees the company's research and development of drugs for ALS and related diseases. Today, on Endpoints, Dr. Fradette joins us to talk about the current state of ALS research, the biggest challenges facing researchers, the developments that give her hope for the future, and how the ALS research community could be better serving people with the disease. Thanks for joining us, Steph. First off, can you just introduce yourself and say a little bit about what you do at Biogen and your history in ALS research?
Absolutely. Thanks, John, for having me. So my name is Steph Fradette. I oversee our neuromuscular development unit at Biogen. So I spend all of my time thinking about ALS, SMA, and other neuromuscular diseases. And I had actually started my career back at Biogen, at least, on the dexpramipexole program. So over a decade ago, and then did a short stint, or more than a short stint, on the SMA portfolio for a while, and then came back to the ALS side of the team, just in time to bring the tofersen program forward. So a relatively long history, nowhere near as long as the vast majority of people working in the space that have been in it for the long haul. But that's a little bit about what I do. We essentially oversee the development, so everything from research to sort of the post-marketing phase, if you will, designing clinical trials and analyzing data from those trials sort of fall within the remit of my team.
Got it. Awesome. And how would you describe what the general state of ALS research was like when you first got started, and how have things changed since then?
Yeah, so even just thinking back to the time that I was working on Dex, so again, over a decade ago, I feel like we didn't know this at the time, but we were operating a bit blindly relative to where we are today. So using Dex as one of many examples in the field, there were data from a phase 2 study, a relatively small, short duration phase 2 study, that were encouraging. And everyone was really excited about what that might mean. And it triggered initiation of a massive phase 3 study called Empower, so over 900 patients in that study. And the results from that study were definitively negative. And I often think to myself, what if we knew today, or what if we knew then, I should say, what we know today, when we were analyzing data from that phase 2 study? I tell anyone that will listen how much I wish we could go back and look at samples from the participants in that phase 2 study to see whether or not there were imbalances in the treatment arms that could have explained the results. So were there individuals in the placebo arm that actually had higher levels of neurofilament, suggesting that they were going to progress more quickly? And if you would have accounted for those differences, would you have actually not seen a difference between treatment and placebo, and then essentially avoided this massive phase 3 study? So I think we're so much more sophisticated today. We have so much more to learn about the underlying disease pathophysiology, and we're still building that toolkit and rebuilding the toolkit that we've had, but we've come so far in our ability to be more objective when interpreting data and prioritizing the right programs into later stages of development.
And, not even just in better ways of understanding the disease, but there are also just more potential ALS treatments and trials currently than, there ever have been. And we've even seen some drug approvals recently, including tofersen, which you worked on. What do you think is driving that progress?
I think it's, it's a number of different things. Firstly, and perhaps most importantly, it's the sheer determination of the field to understand the pathophysiology and to, invest in things like, biomarker development over the past couple of decades. Again, I think we're just now seeing the fruits of that labor with a biomarker like neurofilament, and I'm hoping that we'll see sort of this synergistic acceleration of identification of new biomarkers and endpoints going forward. There's also been, again, for a number of different reasons, sort of an acceptance that our toolkit has been imperfect to date, and that the things that we were using to measure response to therapies was limited. And I think that there's been an acknowledgement of that, and not just an acknowledgement, but a desire and a sort of a drive to do something about it. And so you see, groups like AMP ALS and Precision ALS in Europe coming together to really define the future state by way of that. What does that toolkit look like? What are we actually measuring across these different clinical trials, and how do we sort of get ourselves to a place where we are being more objective and limiting the number of false positive or false negative results? I think there's also been, with a lot of different therapies being in clinic, it's a good thing, but it's also something that we have to strike a balance on. We don't want to just be throwing spaghetti at the wall, if you will. And although one of those things may work in the end, not being focused in what we're prioritizing scientifically can have downstream effects that perhaps isn't the best thing for progress in the field and in patients. And so you see examples like the work that Expert ALS is doing in the UK, where they're essentially looking at a number of platform trial approach where they're using neurofilament to prioritize compounds where they do see a signal to later stages of development. And so people have different ways of going about this prioritization based on scientific plausibility, if you will. And I think that the more of that we see, and the more objective we are, the more progress we'll see on the back end.
Got it. And sort of the flip side of that question is we are still seeing so many ALS drugs that have shown promising results in preclinical or early phase studies fail when they get to these larger late stage trials. And I think you've already said a little bit about this, but generally, why do you think this happens? And what can the industry do to help address it?
Yeah, and Dex, to your point, Dex was a good example of this. I think we have come to learn, and again, I don't want to make a blanket statement because there are nuances to what I'm about to say, but in general, we cannot rely on small studies over short durations when we're looking at clinical outcome measures, because they're imperfect at this stage. And we're going to do more work, and we are doing more work to come up with more sensitive and objective markers. But based on what we have today, when we're looking at clinical outcome measures like the ALS-FRSR, we have to be more cautious about interpreting data from these earlier stage studies. And I think that the field as a whole has, come to realize that, and now we have a number of examples where that has sort of proven true. And so you're already seeing not just people talking about the problem, but also implementing solutions. So as an example, when we're interpreting data from our Taxon-2 trial that read out negatively just a couple of months back, we really anchored that decision on the absence of a reduction in neurofilament. So we're using neurofilament as a much more sensitive way of assessing whether or not the therapy is doing what we need to do in that case. We needed, based on the mechanism, based on the burdensome nature of an intrathecally administered ASO, we needed to see a very robust treatment effect, as evidenced by a reduction in neurofilament. And so that was a critical component of the decision-making framework. We also were able to incorporate baseline neurofilament levels as a way of making sure that we didn't have imbalances that were driving a signal, whether it be, a false positive or false negative signal in that data set. And we are by no means the only ones doing this. It's really happening across the field and across clinical trials. And so I think that we will, there have been a number of different cases of exactly this that have happened of late, but I'm really optimistic that we'll see fewer and far between as we go forward.
And so continuing to focus in on clinical trials, and there's been a lot of talk about things like patient-centric design and just taking the needs of the people who participate in these trials more seriously. So how can pharmaceutical companies in the ALS space make sure that the needs of people with ALS and their caregivers are properly considered when considering all these other factors that go into conducting research and clinical trials like these?
Yeah, absolutely. I mean, I think one thing that I talk about quite a bit is just the importance of seeking input from the community, but not just to check a box, seeking it to really learn from it. And if we take a look back over the past, however many years, the community, people living with ALS, their caregivers, they've been shouting from the rooftops about a number of things. They've been shouting from the rooftops about the inadequacies of our gold standard endpoint, ALS-FRSR. Danielle Boyce and a number of others from the community published on this. And again, we didn't immediately react to what they were saying. And had we, perhaps things might look a bit differently in the field as of today. The importance of biomarkers, the importance of incorporating and including at-risk carriers in clinical trials, and then ensuring that those clinical trials are designed in a way that we are reducing the burden to the best of our ability and providing the support that we need to provide. We can only do that with input from the community. So I think that there are a number of different ways to seek this input. Some groups and some companies will have, patient advisory boards where they're standing advisory boards that provide input on a regular basis. In some cases, coordinated through NEALS and other programs. Wherever or however you go about doing it, I think that it's obviously, goes without saying, it's critical. And again, the more that we can do it, because we really truly believe, and I truly believe, that we don't know what we don't know, rather than checking a box, the more, I think progress we'll make in that front.
Yeah, and can you maybe just say some examples or some specifics of how, ALS advocates have, had a hand in shaping research initiatives and trials that have happened recently and improving them compared to what came before them?
Yeah, absolutely. I think that, I mentioned sort of the work that Danielle and Michael and Kathy and others had done on the ALS-FRSR. I think we're just now seeing people wake up to what that means, and not just putting Band-Aids on the problems that we have with some of our clinical outcome measures, but really getting to the heart of what matters for people living with the disease and their caregivers. And so while we may see some Band-Aids, because we gotta, we have what we have in the near term, I think there are broader efforts to really take this feedback and do something better, create better outcome measures for the community. I would say that when we look at groups like End the Legacy, the impact that group has had on the field in such a short time is remarkable. Really, having such an instrumental voice in making sure that all of us are considering the at-risk community. Again, not assuming we know what the at-risk community wants or needs, but hearing from them directly has been instrumental. Obviously, for us at Biogen, it's critical because we're running an at-risk interventional trial. But I'm often reminded by Jean and others that we really need to be marching towards a future state where it's not just a separate, pre-symptomatic, asymptomatic interventional trial. It's how do we incorporate at-risk carriers into all of our clinical trials where, where the mechanism and the modality makes sense to do so. So I think that they really are, the most important voice at the table. And I'm not just saying that. I think it is absolutely critical. And if history sort of tells us anything, it's that we should be listening even more than we have been to date.
Yeah, and we've talked a lot about the role that people with ALS and advocates can have in shaping research and trials. But beyond that, is there just anything else you think that researchers in this ALS space could be doing better to meet the needs of the people with the disease today and their concerns and things like that?
Yeah, absolutely. I think some of it's hard, right? We know that things are broken, but a lot of us are running trials. So dealing with accepting that things are broken and fixing the things that are broken while you're running a trial that may incorporate some of those imperfect things is a tricky thing to do. But I think we've sort of reached this point as a field where we don't have a choice. And so we're all sort of holding hands on what do we iterate on, what do we put a Band-Aid on and optimize to the best of our ability today, and then what do we do over the longer term to really find ourselves in a much better place 5 years from now. I would say in general, and I've sort of touched on this, prioritization of programs that have the highest scientific plausibility is something that we all know we need to do, but it's difficult. And how do you assess scientific plausibility, particularly when you're thinking about sort of our limited ALS models, particularly for the sporadic population at present. So I think that the more work we can do to understand the pathophysiology of the disease, the easier this becomes. But really, again, prioritizing recruitment and speed in trials where we do have this higher scientific plausibility will get us to answers faster. And we have these monogenic forms of the disease like SOD1 and FUS, where we understand what's wrong, we understand in principle how to treat it, and we can learn so much from those programs, as we have seen in both cases, that can influence how we're managing the broader ALS population and trials in the broader ALS population. So I think that's critically important. I think being objective when we're interpreting results from trials is something that is absolutely critical. Being willing to end a program when the data suggests that it doesn't work so we can move on and prioritize the next program that, again, it does have higher scientific plausibility. These are things that can be difficult. We all get attached to our research. We all get attached to the programs that we support. And so being objective and holding each other accountable to be objective is something that I think we're all working towards together.
Awesome. And once again, I think a lot of what we've talked about already probably alludes to this, but is there anything specific that you can look at, especially about how things have changed since you started working in the ALS space, that gives you hope for the future of ALS treatments and research?
So much gives me hope. I think that, again, just taking a trip down memory lane with dextromethexel, when that phase 3 study read out negatively, Biogen, and I know other organizations in the space, really made the decision to double down on genetic forms of the disease, where we really had a better understanding, as I mentioned, about what was going on and how to treat it, anticipating that we could leverage those learnings in developing therapies for the broader population once sort of we understood them better. And that's really panned out. And so I think it took time, but we are at this place where we're able to isolate some of the variables that have been in play over time and really now start moving more quickly when it comes to testing and understanding whether or not these therapies have potential. I think that the commitment to developing novel endpoints that not just assess the potential for slowing of decline, which is essentially what our toolkit consists of today, but also thinking optimistically about a future state, endpoints that can really measure improvement if there are therapies that are able to actually help individuals regain strength and function that they previously lost. That might be something that we need to assess with a clinical outcome measure that doesn't exist today, or exists but we're not using it routinely in our clinical trials. So I think that I'm optimistic about the work that's going into sort of the endpoint development space. I'm optimistic that we're going to sort of leverage the work that the AMP-ALS and Precision and other groups of that kind are doing to progress biomarkers. Obviously, the work that's happened on the cryptic exon space is remarkable and so encouraging. Some of the most promising targets exist and modalities exist because of the work that has been done by the academic community in that regard. So I think we're just approaching this point in time where we have a better understanding of the pathophysiology, a better understanding of how to run objective clinical trials, and I'm quite optimistic about the next 10 years.
Awesome. So just to follow up on that, actually. So I'm sure this is pretty clear, but I just want to dial in on it a little bit, I guess. So you're saying when you say the broader population, the hope is that like things we're learning from these treatments that target forms of genetic ALS could end up teaching things that could be applicable for specifically the sporadic forms of the disease, right?
Yes, exactly. Neurofilament being a perfect example, where, we may not have implemented neurofilament as a marker of treatment response absent the experience in these monogenic forms of the disease, if you will. And so that's a learning that can be pulled into the broader population as well.
Awesome. And so getting to the end of the questions I have here, we've talked a lot about the ALS research space from on high, looking at everything. But this is an ALS Therapy Development Institute podcast, so I would be remiss if I didn't ask you to comment on, looking at this ALS research landscape. What do you think? How would you describe ALS TDI's place in it, and how are we sort of helping moving forward some of the trends that you've talked about?
I'm so glad that you asked this question, and I'm not just answering this way because I'm talking with you. I think the world of ALS TDI. I think, Fernando, Nadia, Carol, they have a voice and reputation that is so distinct and so powerful to the community. They're a sounding board for me, and I'm sure many, many others. Fernando is one of the first people that I, call when I am contemplating or struggling with a scientific question, and has been over many years. So I think just the resource and the people that work at ALS TDI, it goes without saying, are among the best and some of my favorite people in the world. The ARC study, I still refer to it as the precision medicine program sometimes, but the ARC study, I think, is a beautiful example of what needs to happen on a broader scale. And I know that ALS TDI is part of sort of the AMP initiative, but really collecting these data, the data commons tool is beautifully done, and we can learn so much. We already use it on a regular basis at Biogen, and I know there's a lot more work happening to look at things like novel biomarkers and such, from the ARC participants that will further inform the work, not just on neurofilament, but also on other exploratory biomarkers as well. So in addition to sort of the scientific work that the researchers at ALS TDI are doing, which is, profound and so encouraging, I think, the ARC program, the Trial Navigator website, there's so many things that the organization is doing that I think are meaningfully supporting the field and the community. It's pretty unique.
Awesome. And can you talk a little bit about how you see different organizations sort of throughout the space collaborating, and how that collaboration is important in sort of reaching, once again, some of those things that you've mentioned that make you hopeful for the future?
Yeah, I truly believe that the ALS community and field is unique. And I always say, I hope it stays that way. It is distinct from other disease areas, and it tends to be just beyond collaborative. The number of times that, industry partners are having open conversations about what they're learning in real time so that we can incorporate those learnings in our own trials, it's just common practice. And there's such a close connectivity between the academic industry and patient advocacy communities. And I think there's a recognition, and it's becoming increasingly the case, that there's a recognition that none of us can do what we do without one another. And so really seeing, there are very few, and there should be none, sort of situations where you have a setting where you don't have all of those different groups represented, where you're having a scientific discussion at a congress. Historically, it has not always included, these congresses have not always included people living with ALS or their caregivers, and that's changing. It needs to change more, in my opinion. But I think that having all of those different groups at the table is, like I said, unique and so critically important, because we all have blind spots and we're all learning from one another as we go. It's inevitable that everyone, again, is protective of their own thing and their own research. And I think that efforts like AMP-ALS are really forcing us out of our siloed work. Even though there's a desire to do that, I think it's really, again, putting it into practice, where siloed data sets on natural history are powerful, but consolidating all of those data into a single knowledge portal is, exponentially more powerful. And so I think that the collaboration is happening, and it's critical, and I hope we never lose it.
Awesome. Well, yeah, thanks so much for joining us.
Thanks, John.
There are currently no treatments to stop or reverse ALS, but the ALS Therapy Development Institute is working to change that. To learn more about ALS TDI and our research to end ALS, visit als.net. Thanks for listening.